huge decision that gets made in clinical labs every single day, one that really matters for patient care. When you've got a potential infection, how do you figure out exactly what it is? Which tests do you run?
You often hear this framed as a big showdown, right? In one corner, you have the classic old-school champion, traditional culture, and in the other, the flashy, high-tech newcomer, molecular diagnostics. So who wins the belt? Who's the real champ of the lab?
Well, here's the thing, and it's kind of the whole point. It's not a competition. Not at all. Actually, the best labs out there know these two aren't competitors. They're teammates. They have completely different skills, and the real secret is knowing exactly when to put each one in the game.
So, to get a handle on this, let's break down the playbook for both of them. And we've got this amazing guide to help us out, complete with these two little microbe characters who are going to walk us through how they each work, their biggest strengths, and most importantly, when they get the call.
Okay, first things first. To really get when to use them, you've got to understand how they play the game. So how do these tests actually do what they do? Let's start with the veteran, the old reliable, traditional culture.
I want you to think of traditional culture as being kind of like a gardener. For real, you take a sample from a patient, you plant it on this special nutrient-rich plate, and then you wait. You're basically giving any bacteria in there the perfect little environment to grow up into colonies you can actually see. Once they've grown, you can hit them with different antibiotics to see which ones work. It's incredibly thorough, but as you can imagine, it takes a little time.
Now, if culture is the patient gardener, molecular is the high-tech CSI-style detective. It doesn't need to grow anything, no waiting around. Instead, it uses a powerhouse technique called PCR to go straight for the genetic fingerprint. It finds a tiny piece of a microbe's DNA and, like a genetic copy machine on hyperdrive, it makes millions and millions of copies. It's kind of like a super fast barcode scanner that just reads the pathogen's unique code, and bam, you've got an ID in just a few hours.
So we've got our gardener and our high-tech detective. Now it's time for the scouting report. Let's dig into the specific strengths and weaknesses, the pros and the cons that every lab and doctor has to weigh. And this is where it gets really interesting, because you can see the trade-offs. Culture is still called the gold standard because it proves the bacteria is actually alive and causing a problem. And its killer app really is the full antimicrobial susceptibility test, or AST. That's the detailed report that tells doctors exactly which drugs will work. Plus, it's pretty cost-effective. The downside? It's slow. We're talking 24 hours, maybe even days. Molecular, on the other hand, is built for speed and sensitivity. It'll find a bug in hours, even if there are very few of them or they're already dead. The catch? It's more expensive, it can sometimes be too sensitive and pick up leftover DNA from an old infection, and it doesn't give you that beautiful, detailed AST report.
Okay, with all that in mind, we're at the most important part, the actual game plan. In a real-life situation with a real patient, how do you make the right call?
The strategy becomes pretty clear when you see it laid out like this. Culture is your go-to starter for most routine infections, where you have a little bit of time. You use it when you need that full susceptibility panel to nail down the right treatment, or if you're just monitoring to make sure a treatment is working. But you call your specialist, molecular, off the bench for the really urgent stuff, like suspected sepsis, where every single hour matters. It's also the MVP when a patient has already started taking antibiotics, which can stop a culture from growing, or for those tricky cases where the culture comes back negative, but you just know something's up.
So after all that, what's the final score? What's the one thing you should remember from all of this? Let's boil it all down to the absolute core difference.
I mean, how great is this? It's just so perfect. Culture answers two questions. Is it alive, and what can we use to kill it? Molecular answers one question. What is it? And it gives you that answer fast. That's it. That's the whole ballgame right there. So simple, but so incredibly powerful to remember.
And that brings us right back to where we started. This was never a versus situation. These aren't rivals. They're just two powerful, specialized tools on the exact same team, working together to give patients the fastest, most accurate diagnosis we can. Which leaves us with a pretty cool question to think about, doesn't it? As our technology just keeps getting better and better, with faster sequencing and way smarter AI, how is this partnership going to evolve? What new plays are they going to add to their playbook? I don't know the answer, but it's definitely going to be fascinating to watch.